Welcome to Convergence. My name's Michael Doran. I'm a California attorney. Albert Einstein once stated that among the most difficult problems in all of physics is the Earth's magnetic field. As an attorney in California, I'm going to argue that the Earth's magnetic field is actually one of the most difficult problems in all of biology. Albert Einstein certainly thought about this in terms of what life on Earth could teach us about the physics of the Earth's magnetic field. In fact, he had a friend who is a beekeeper, and some of the behaviors of bees are, at the time that he wrote this little letter here I'm going to read to you, were even then known as having interesting properties. And obviously the name of this show is Convergence, and it's about how technologies and information have advanced, and then they converge on each other and they tell us things. But when Albert Einstein wrote a letter about this, he wrote, "I'm well acquainted with Mr. Fritsch's admirable investigation." He's a beekeeper. "But I cannot see a possibility to utilize those results in investigation concerning the basis of physics. Such could only be the case if a new kind of sensory perception respecting their stimuli could be revealed through the behaviors of the bees. It is thinkable that investigation of the behavior of migratory birds and carrier pigeons may someday lead to an understanding of some physical process which is not yet known. Sincerely yours, Albert Einstein." Today, I want to go back in time to the beginnings of life in order to then comment on what we do know about bees and birds and, say, salmon and other life forms that use Earth's magnetic field to move and to behave. And the best way I can explain why this makes a tremendous amount of sense to do it this way is something that I thought about at the time when it was occurring, when the great chess champion Kasparov played a supercomputer called Big Blue. This is the early 2000s. And what was fascinating was how he was able to beat the machine. Big Blue was a supercomputer, the fastest computer in the world at the time. So the way he explained that he was able to play the supercomputer and win was he knew what the basic programming of the machine was. So for instance, the programming had a rook being worth 5 points, a bishop being worth 2.5, the knight being worth 2.5, the queen being worth 10, and then the pawns being worth 1 point and a little bit more as they advanced towards the back wall, where they could change into another piece. And so what the computer would do was there was a formula to calculate what is the most points that a move could bring the computer. And therefore, in terms of a program, that would lead to the best result for each move. And that was the program that Kasparov knew he was going against when he was going against the computer. Now, the interesting thing is that's not the object of the game. The object of the game is to put the king in checkmate. So Kasparov was using a different way to get to what he was trying to do than what the computer was. So even though the computer had this ginormous advantage to get the most points through calculation, Kasparov had a handle on the game and was able to beat the supercomputer. And this is, by analogy, what we're trying to do when we're talking about life and what life's basic programming was. Is it formed? The abiogenesis discussion, and then therefore how we can use that basic programming to understand what is going on now. So it's kind of like that joke, an amoeba is a simple life form, now try to make one. And so what's funny about that joke is an amoeba is incredibly complex, and it presents the same kind of problem where if it's a calculation of huge time frames of evolutionary processes, there's just numerous calculations and transactions that occur to get to the amoeba, and so it's easier to describe those processes and what those processes were doing in order to understand really what an amoeba is. And so we have to start from the beginning, which is that the Earth's magnetic field is a biological problem that is the fundamental basis for feedbacks from life. And that is because if the Earth's magnetic field is too strong, then it attracts gas and other things, space dust, space water, space hydrogen, and soon you get a gas giant like Saturn or Jupiter, and those are dead planets. They have storms like the red dot on Jupiter that have gone in the same place for hundreds of years. It's incapable of being dampened. Or you get a planet like Mars, where the solar winds and so forth have stripped away all atmosphere in the absence of a strong magnetic field. So we have an implausible complexity with the Earth's magnetic field. It's a sort of a Goldilocks. It's not too hot, not too cold, it's just right. And the only way that you get to that point is through life. And that's the fundamental problem that life had to first solve. And then, therefore, that tells us what the basic programming is and what's going on. What's going on that evolution over time has calculated the best moves, and then eventually you get something very complex, but it doesn't tell us what the basic programming is. We have to go back in time when first life was created, and what was the problem being solved? What were the conditions? So the first thing that, in terms of what's converging, what technology has come about that's given us a better picture than from when Einstein wrote this letter about bees and migratory birds and so forth, that they can't tell us anything because we don't know anything yet about the processes, but now we do. So what has occurred? Well, one thing we know is what DNA and RNA are. And in fact, there's new research that indicates that RNA can spontaneously grow in an ice crystal, which gets us right there in terms of where I believe these beginning processes occurred, and that is that RNA could be both a feedback and a model at the same time, and from there complexity is calculated. And so I want to talk about what I believe are the two, what I believe are the basic programming of what RNA was at the beginning of when we go from chemicals that really we don't consider to be life to say an amoeba or other life that we know is life, and we think of it as life. So you have this boundary between chemicals that we don't think of in we can make them in a lab, but that we don't think of them as life, to what we have today, all the way up to what a bee is, so that we can understand what properties of, say, a bee that reflect and can tell us a story about what's gone on as life has become complex. So the first thing I want to talk about is just simply what we know about DNA and RNA. RNA is a double helix, and it's full of so we know what it looks like and what it's made of now. And if we go back in time to when the Earth was full of just nucleotides, and they were available, and they would be in the ocean, the building blocks of them anyway. And let's say that the building blocks got blown up into a storm, into a cloud droplet that was supercooled, that was starting to freeze. And what we know about this, what the sun does, is that the sun has different types of outputs, like X-rays, like solar winds, and then on the backside we have cosmic rays, and different things that would either cause there to be a source for water, like hydrogen particles from solar winds, or would be energy that would actually strip the atmosphere away if there wasn't a magnetic field. And we also know that the early Earth was bombarded by rocks and asteroids and bollards, and was molten and had a metal core to it. So it probably had some magnetic field. Now, we know that if you had a droplet of water, and in that water you had a nucleotide, it will experience potentially DC fields, and will also potentially experience resonances, depending upon how close lightning strikes were. So let's talk about that a little bit. So let's say you have a field, it's positive and negative, and then you have your droplet inside it, and inside the droplet you have your DNA. So you're going to have, on the one hand, you're going to have a field, and then on the other hand, every time that lightning strikes, you're going to have, very much like if you took a metal bell and you struck it with a hammer, it's going to cause the Earth's magnetic field to resonate, and you're going to get Schumann resonances. And really close to where that bell strike is, you're going to get higher frequency resonances. You're also going to have from the sun, you're going to have really super high frequency light called X-rays. And an X-ray, it's, if you had a picture of say an atom, an X-ray, its wavelength is about the same as the length of an atom. And because of that, an X-ray is going to make it hard for molecules to be stable because the energy is so great that it gets into the bonds of the chemical. So you had all this going on in the early Earth. And what's interesting about the nucleotides is if they replicated in mass, they began to potentially solve the magnetic field problem in terms of lightning produced and the global electric circuit, which then in turn affected the Earth's magnetic field. And so you have this feedback. And not only do you have this feedback that's potentially affected by the nucleotides, but that solution that the nucleotides might have initially randomly solved in some way became a model and then would get down to Earth and re-replicate and maybe replicate in such a way that is mass produced, I guess is the best way of saying it. So then that solution gets reproduced collectively with the nucleotides, and it becomes more complex over time. And so if that's our basic programming, then there's some interesting conclusions that we can go from there about life now. So the assumptions that I'm making are that the nucleotides were a solution set and a feedback, and then over time they this solution set became more complex. RNA in particular, which a recent study found could self-replicate in ice, is probably where it started. The RNA came before the DNA and then went to DNA. What's interesting there is what the letters stand for. R and acid. And this is where you start to get into the phase change rates. So what happens is, if you have an acid, whether it's carbolic or carbonic acid, where carbon life forms, or RNA, RN acid, all these different acids, when you get into a DC field or a positive and minus field, and you have a water droplet that's super cooled, and in there is an acid, the phase change rate is going to be affected by the strength of the field and the amount of the acid that's in it. And that's because the migrations that occur of the acid in the ice crystal changes the morphology or the shape. It'll give it a Christmas, for instance, it could give it a kind of a Christmas tree shape of the ice crystal forming. So it changes the phase change rate of the ice crystal. And it does so in a calculated way, depending upon the size, shape, mass, and charge of the nucleotide. Or now with the carbonic or carbolic acid, with the carbon dioxide, you're going to have an acid that is weak and not necessarily weak, but you're going to have an acid that doesn't change. It is valuable. It can cause the phase change rate to occur, and can be used to dampen the cloud behaviors and organize the clouds. But it can't do so in a way that's a model. It can work symbiotically with nucleotides, but the nucleotides are what gave it its feedback, its ability to replicate and then come back and be a model of part of a global solution set to a living Earth and the Earth's magnetic field. I know I'm all over the place with this, but so that's with the DC field. Now, this is where it gets even more complex when you start talking about quantum mechanics. It's like, how in the world do you get quantum mechanics out of this? Well, So you have your DC field, right? And you're going to have that in any thunderstorm. You're going to have a potential difference. You're going to have these droplets that are exposed to a field. And again, if the supercooled droplet has R and A in it, or it has this acid in there, and it's affecting the morphology of the shape of the frozen droplet, it's very much like electrophoresis. So electrophoresis is how scientists now are differentiating different samples of DNA. It's like your sample of DNA is unique. And that's how, from a statistical standpoint, they look at the outcome of electrophoresis, and they look at they see how you have a unique sample, and then you compare it to another sample, and this is how your DNA is matched up, how different studies, probabilistic studies, take place today. It's electrophoresis. And what that is a sample is put inside a gel, and again you have a positive and you have a negative, and this gel is in a strip. And in the electric field, the nucleotide begins to migrate and separate themselves out, because they're going to move depending upon their size, their shape, their mass, and their charge. And the movement then results in your sample of genetic material having these bands, because the movements depend upon the friction that you're going to see from the gel, as well as what the DNA looks like, how it moves through there because of how it looks like. And then a color agent is put on the nucleotides, and you get these bands which tend to be unique, unless you have the same source of a sample, then you're gonna get a match. And it's very telling as the studies have become more robust about what DNA is, and what different types of DNA, that different creatures or different functions that a life form has. We're learning all kinds of things from these DNA studies. Well, that's just with a static field, a positive and a negative. Well, the next question is, what if we have an alternating current, an AC current? And an AC current is exactly what you would get from space, from the sun. So for instance, whether you're talking about an X-ray, and an X-ray from the sun isn't going to penetrate the Earth's atmosphere now. But that doesn't mean that during the time when life first formed, that an X-ray event would not reach the Earth. It's hard to tell at this point. Let's just say that it's unknown. But certainly the resonances were different then. And that is, today when you have a lightning strike, we have Schumann resonances that are very observed. It's all textbook stuff. But it's hard to say exactly, as life formed, what was going on in terms of the type of resonances that there were. But there certainly were resonances, and we know that early Earth did not have as much oxygen in the atmosphere, and therefore ozone would have been different. Levels of ozone would have been different. Obviously there's some things that were the same, but different levels of different chemicals were different at a different time as life has come into play. That obviously what the atmosphere is made of has changed, but for sure you would have had alternating currents too from lightning that existed in the beginning of the formation of life. And we know that it's about 3.7 billion years ago. It's a long time. And you had free nucleotides. And So what would happen with the nucleotides when you had an alternating current? And I believe this is where the quantum mechanics becomes interesting and important, and where we can talk about what a quantum brain is, and where we can talk about how the nucleotides had a quantum process. And that is, if just some basic quantum mechanics, if you have a wave hitting an atom, let's just say a hydrogen atom, and it's a high enough energy, it can basically move where the electron is supposed to be. And if it can move the electron, then it's going to move where the proton is supposed to be. And this is how many biologists think that you can have mutations, is because when you get these, when you get electrons being in different places, and there's a probability that they're in different places, then you have a probability that, not a absolute, but you have a probability that a proton is going to be in a certain location as well as it is affected by the electron. And the nucleotides are just chock full of hydrogens, whether you're talking about the 4 base chemicals or not. All of them have hydrogens, and the nucleotides themselves are in a spiral or a double helix that it's themselves look like waves. So that if you had the right frequency wave that is occurring in conjunction with the base and the chemicals that go with the nucleotides, you're going to get potentially not only this solution set of how fast a droplet of water that's super cool freezes, and therefore the cloud behavior, and how that is a calculative response to an input from space, but also you have this ability of the nucleotide to actually change, and by being changed, it will affect not only how fast it goes through the gel, and what pattern, what actual DNA you have there, or RNA that you have there, but it becomes a quantum machine to solve, depending upon circumstances and how life became more complex, the type of answer that was produced and then mass produced later, as life went through this cycle of being a solution set. And then if it rained down onto where chemicals were available in the soupy seas of that time, you could get a mass production of nucleotides, and then the next surface though that comes will pick it up, and it gets amplified in terms of how it's answering the stimulus from space, and then you get a feedback of the nucleotides, and from there life becomes more complex. You get a symbiotic relationship between cells that are calculated and formed, and they're able to contain chemistries. They're not able to do the same thing in clouds like the nucleotides could. So you start getting this connected relationship between CO2 or carbon, we're all carbon life, and then this connected relationship with cells. And so the chicken or the egg question is really is neither. It's the male part. The nucleotides came first, that calculated what is eventually became the egg, and then eventually became the chicken. So the AC part is then really interesting, because this is the start of the quantum brain. And what it's telling you is that as